Thank you everyone for joining today. For those who don't know me yet, I'm Alex Lemens, the director of our Molecular and Genomics Informatics core. We also have on the line Pat from the director of our Genomics Center, and really the one who manages all of the single cell projects that we have. But then we also have the team from S2 today who will be presenting on their technology that really will help with a lot of single cell preps. Considering how critical that the quality of the input samples is, I'm looking forward to learning more about their platforms. That way, something that we could possibly introduce going forward. But before I turn it over to them, just the general housekeeping items. If you have any questions, please drop them into the chat or the Q&A box. We'll monitor it as we go, and any questions we can answer on the fly, we will. Anything that's not mentioned during the talk, we'll bring up at the end. So with that, I'll turn it over to the S2 team to learn more about their platform.
Awesome, thanks. So yes, my name is Nathan Pereira, so I'm a co-founder and field application scientist at S2 Genomics. And like you were just told, I'm here to discuss the Singulator product family, kind of for automating tissue dissociation. And we'll kind of explain the benefits of an automated platform and then kind of go through some data sets generated by us and some of our customers. And I also have two of my colleagues here with me, Hannah and Sarah, on the sales and marketing side. So at the end of the presentation, if you have any questions for any of us, feel free to let us know. So what is the Singulator 100? So the Singulator 100 is an all-purpose platform for generating single cells and nuclei upstream of a variety of applications, whether that be mostly single cell and single nuclei RNA-seq, ATAC-seq, site-seq applications, a variety of genomic applications, but it's also helpful for a lot of cell biology applications. So we have lots of customers who isolate and purify cells for FACS sorting, immuno-oncology applications, opening up plenty of opportunities for, in the future, personalized medicines, vaccines, culturing cells, culturing organoids. So a wide variety of applications. The main problem, as I'm sure many of you know, with manual cell isolation is that it's not only time consuming, but it's very difficult. Lots of the people that I work with, either have a manual protocol that they've developed over lots in trial and error that they're pulling from papers, and it requires skilled experts. It's really time consuming to develop these protocols. There's no standard place to go and get them. And then the big thing is that a lot of these manual isolations can have technical bias in terms of cell type representation, in terms of each person that does the isolation. Each person can do things slightly differently. So kind of the benefits of an automated platform are kind of standardizing the workflow, so you know what you're going to get out every single time. Similar for nuclei, requires skilled experts. It's time consuming, low quality, variable yields. So any sort of sample preparation artifacts that you have going into your data is going to lead to data artifacts coming out. So this red circle, part is kind of the last part of this single cell, single nuclei workflow that's yet to be automated, and that's what we're aiming to do and what we're doing. So we feed into single cell processing pipelines, library prep. We've generated lots of data with various companies: Parse Biosciences, 10x Genomics, Mission Bio, Fluent, to name a few. And that's what we're working on doing. So we have two products out right now, the S200 and the S100. Here's a picture of the S200. It's basically a completely automated cell and nuclei isolation. So on the S200, the new version of the machine, we can do two samples at once, and that can be one sample of nuclei at a time, or one cell at a time, two nuclei, one cell, one nuclei. So they're completely two independent bays, and they're completely customizable processes. So I like to say I can usually help port a manual protocol onto the Singulator pretty easily with a little bit of optimization. It's an open reagent platform, so we provide our own cell reagents, enzymatic lyophilized reagents for a variety of cell types. And we have our own nuclei reagents that live on board in a chiller kept cold. So basically the machine is ready to go, walk up to do nuclei isolations and prepare enzymatic reagents beforehand. But it is open platform. So if you have a golden nuclei isolation buffer that works really well for your specific tissue type, you can totally load that on the Singulator as well, and run it with that. Same thing with a special enzyme solution. It's temperature controlled. You obviously want to do nuclei as cold as possible. So we use cold cartridges, and the machine is cooled to do all of that at about 0 to 4 degrees for C. And then cells, you have is traditionally 37C, but you have the option of doing that cold as well with cold dissociation reagents or at room temp. It's fast as well. Nuclei on the S200 is about 6 to 7 minutes. There's a variety of different protocols that can be longer, for more robust and more lysis. And cells can be as quick as 25 to 30 minutes, with some being, a little bit longer as well. Skin, some more difficult to dissociate to sample types can be up to an hour. It also is pretty robust for small sample sizes. So we just released about 6 months ago an updated nuclei isolation cartridge plus that goes down to 1 milligram of tissue. And that's been being used, in a variety of frozen tissues down to 1 milligram in size. So again, the goal of this is to simplify and automate the workflow. So just kind of overall, your tissue will go into our cartridge on the top. We have we have cell and nuclei isolation cartridges. I won't go too much into the details, the differences between them, but they have different disruption methods and then different filtration units. So samples are disrupted in the cartridge, washed, and then filtered in the cartridge as well. So you have at the end of the run have a filtered and washed suspension that you would then take and centrifuge and kind of work towards your downstream applications. Nuclei, again, as I mentioned, about 5 to 7 minutes, and then you'll take the sample out of the cartridge, centrifuge it down, and resuspend it, usually in our storage buffer that we provide, our wash buffer, and then you can go, with downstream applications. Some tissues may need additional cleanup, density gradients, that kind of thing for example, like brain tissue, you need to get rid of myelin debris. For single cell suspension, same thing. After the enzymatic digestion and dissociation, they come off the machine, they're centrifuged. Usually an RBC lysis is done, and then another clean out of the RBC lysis, and then you're kind of good to go from there. Again, depending on the sample type, you might need some sort of additional centrifugation or washing, but usually that's the entire workflow to go down towards downstream applications. So as I was mentioning, we have a wide applicable range of sample types and sample tissues and sizes. So this is some data from as I mentioned, our new NIC Plus cartridge, going down to about 1 milligram of tissue. This is showing, I believe, nuclei yield data per milligram from frozen brain, frozen kidney, frozen spleen, liver, and heart. And you can see on the sample sizes from about 0 to 30 milligrams, the robust, reproducible isolation yield is pretty reproducible and pretty much the same across the sample types. We do see a little bit of drop in yield per milligram as we get down to about 10 to 1 milligram, but it is, it's robust down to 1 milligram of tissue. So again, some of the benefits of this automated tissue processing is it's going to improve reproducibility, which is inevitably going to reduce time and cost. You don't have to sit and train people on these, difficult and costly isolation methods. It outputs the single cell and nuclei suspensions ready for processing. It's really quick. I have customers who spend 2 hours doing very intense nuclei isolations that revolve around multiple downsync, multiple incubations, lots of procedures, and they're able to shorten it down to a 10 to 15 minute run if applicable. It's easily optimizable, which I'll kind of talk a little bit more about in a bit. There's lots of different parameters you can play around with. And it works well for flash frozen, fresh, and OCT preserved tissues. So, and one of the other taglines, I like to say is that it's the same process for most tissues. So a lot of people who try the machine will do this exact same nuclei isolation procedure or cell isolation procedure as they would for, honeybees, lots of obviously mouse and rat work, and human work. But we've worked with a variety of interesting tissues, rockfish, lots of insects, plenty of different species. So here's some data kind of showing the high yield and high viability of of various tissue types. This is all generated using our S200. So on the left is the cell isolations. We have our standard tissue types: liver, intestine, lung, spleen, and kidney. And those are the ones that we sell standard lyophilized enzyme reagents for. Those are usually suspended in some sort of media. We usually recommend DMEM or HPSS, and then those are loaded onto the Singulator, usually for cells, minced tissue. And we range from getting about 12,000 cells per milligram in liver all the way up to 600,000 cells per milligram in spleen. And nuclei yield fairly similar, obviously a little bit higher than cell yield, but from a variety of tissues, very low end of muscle and heart being about 4,000 to 5,000, all the way up to high end of kidney, 200,000 to 300,000 nuclei per milligram. So as I mentioned, one of the benefits is the customizability and the flexibility. So you can customize plenty of different options: the dissociation time, the dissociation temperature, mixing parameters, and basically the intensity of kind of these disruptions. And going back to what I said earlier, I can usually easily port over a manual protocol with a little bit of optimization through some sort of customer support. And lots of customers can really do it on their own. So if a manual protocol is incubate in some special enzyme solution for 45 minutes, downsing or mixing every 15 minutes, and then some sort of mechanical disruption, most procedures can be pretty easily ported onto the Singulator with a little bit of optimization. So you can change basically the intensity of the mixing and intensity of this disruption. And then you kind of base that off of pictures from viability data from staining or sequencing as well. It's customizable for the nuclei as well. Nuclei is really, like I said, is pretty standard. Lots of people just use our standard profile. We have a variety of different isolations. I said earlier, it's about 6 minutes. And just kind of briefly, we deliver lysis buffer, do disruptions, and then filtration. There's protocols where we do onboard incubation as well, excess disruption cycles. And it kind of has to do with the more difficulty of isolation of nuclei from certain tissues. Dense tumor types, we have extended, more laborious isolation protocols, and delicate tissue types, we have the slower speeds and less intense disruption cycles. And then something we're rolling out as well for the nuclei protocols very soon is customization of volume. So again, all these runs, all these downstream applications can be really expensive. A lot of the expense comes from obviously the sequencing, but obviously RNase inhibitor. So we're enabling the ability to change volumes to kind of save on a lot of those reagents as well. And we can also provide our own RNase inhibitor for these reactions as well, which we've tested and has worked just as well as most of those being used for downstream single nuclei sequencing applications. So one of the things I've been touting again is the ease of optimization. So here's some data showing kidney cells that were isolated on the Singulator. You can kind of see a pretty poor cell type representation. This was our V1 protocol before launch. Pretty much really just an isolation of proximal tubules and distal tubules and a couple macrophages. So we're missing, a fair amount of cell types. This tissue was used, we put in whole tissue, incubated for 25 minutes without getting too much into the protocol. But we decided, after doing some research and talking to some customers, to change up the protocol a little bit. And this is after a couple runs, our V2 protocol. So we do manual mincing, increase the incubation time a little bit. Again, played around with the settings and agitation style. And And here's the new sequencing data. You can see pretty much all of the expected cell types, proximal tubules. Now we have lots of epithelial cells, even sensitive podocytes and rare podocytes, and a variety of obviously different immune cells. So it's super easily optimizable. This is changing two parameters, incubation time and adding a disruption step, to easily optimize your protocols. So in addition to reproducibility, there's also high quality. So here's some pictures of DAPI stained nuclei. This is courtesy of Aki Minoda Lab at the Riken Institute in Japan. So you can see the intact heterochromatin and the nuclear membranes. This is from a variety of frozen tissues, mouse heart, mouse small intestine, mouse liver, and mouse brain. And a lot of this was also used for ATAC-seq studies. So this comes from a group at Stanford University in California. They were doing OmniATAC protocol on diseased intestinal polyps. And they were gracious enough to test our system against kind of the gold standard OmniATAC protocol. And what they found was, you can kind of see in J, the Dounce and S2 methods overlap very nicely. In K, you can see again the S2 samples cell type representation matches very well with the manual method. And then in L, you can see the differential peaks between the stem cells were nearly identical, indicating the accessible regions of the chromatin were nearly identical between the samples. So it matches, they found that their manual method matched very well to the Singulator method, which was, great for us and great for them. So we also have done work with Mission Bio. So that they were, this was kind of a pilot study, able to use the Singulator for isolating nuclei for DNA sequencing. So they were able to isolate 38 samples from 16 different pancreatic adenocarcinoma patients. And they basically demonstrated this robust workflow. They were able to actually isolate and freeze the nuclei for downstream processing, which they were not able to do before with their manual method. And, opening up whole targeted genome sequencing is vastly important for the discovery and development of precision medicine. So I talked about ease of optimization on the Singulator. So it's also reproducible. So here's a little data set that I'll go through. This is going to be from lung cell isolation of 3 different patients. So these are 3 different lung resection samples from 3 different patients. Unfortunately, they were processed about 18 to 36 hours after isolation, kind of a range of time. And these were all processed using our mouse lung isolation protocols and reagents. So all of our mouse reagent protocols and mouse reagents and mouse protocols in the Singulator are pretty much portable for any of the species, is what we're finding and what we expected. We just do most of our baselining and most of our testing on mouse in-house. That's getting fresh human tissue is difficult. But you can see here the viability was robust across the 3 different patients and across the 3 different isolation and processing times, all above 90%. And when we look at the clustering data, comparing it to azimuth, we see all the expected cell types. The one decreased cell type we do see is basal cells compared to the reference set, which is not too surprising, as they kind of reside in the upper airway epithelium and kind of and have diminishing numbers down into the bronchial, which our samples were deep lobe resections. But pretty nice match to the azimuth data, both AT1 and AT2 type cells, and plenty of endothelial capillary cells as well. So again, here's the cell type representation for each of those 3 patients, and the clustering. So you can see after and before before integration, the nice overlap of the cells, and we have all the similar and all the same cell types between all the different patients and all the different cell isolations. So one of the other things I really want to talk about is kind of nuclei versus cells. So the Singulator obviously does both, but it's kind of going to be up to research application and kind of needs of the lab and abilities of the lab. There's different applications where cells are better and nuclei are better. If you're looking for rare genes, or you kind of want higher genes per cell in your sequencing, cells are going to be a better idea. Nuclei are going to be better if you're having difficulty isolating cells, if it's a difficult cell type or delicate cell types. If you're looking for cell surface markers or site-seq, obviously you need to do cell applications. And then nuclei has some benefits over cells in just obviously throughput. Lots of these cell isolations can be upwards of hours, and lots of hands-on. Nuclei are going to have usually higher yields per milligram. You're just kind of lysing and taking all those nuclei out. And you also see better cell type representation from nuclei, again, mostly due to lots of cells being difficult to isolate and kind of dying throughout the cell isolation process. Immune cell profiling, again, cells, or if your application is not really sequencing based, and you're doing pull down or isolation of specific cell types, well then, yes, obviously cell isolations are going to be more important. So one of the things I kind of like to touch on is kind of this idea of cell type representation. So this comes from a group at the University of Korea in Seoul, and as I'm sure a lot of you know, cell type representation can be an issue for single cell sequencing, especially over single nuclei sequencing. The enzymatic dissociation required can induce cellular stress response and alter the transcriptome, and again, lead to decreased isolation of delicate and more difficult to isolate cell types. So here's some data generated from the Singulator. Here's some mouse lung nuclei. You can see, again, through the QC data, plenty high of feature RNA and pretty low mitochondrial contamination from these samples. And what you see in the clustering data is a pretty wide variety of cell types. So all the expected cell types, the alveolar type 1, type 2 cell types, all the different immune cells as well, plenty of endothelial, clubbing globule cells. So we see, a pretty nice representation of cell types in the mouse lung nuclei data, and pretty low contamination as well. In the cell data, we do see all the expected cell types again, endothelial, epithelial, alveolar type cells again, muscle cells. You can see higher feature count RNA as expected, but what you do see is we do see an increased percentage of T cells and B cells, and again, that's going to be due to the ease of isolating immune cell types over the more embedded, more structurally embedded endothelial and epithelial type cells. So when we take a look at the comparison of the cell types between these two samples, you can see, again, in the nuclei, we see a more kind of equal population compared to the cells. Again, more T cells, B cells, and kind of club and goblet cells in the cell population, and a more kind of even representation of the different types of cells in the nuclei sample. And here's just a graph kind of saying, again, immune cell representation versus non-immune cell representation. So in the nuclei, we're definitely capturing more of those different types of cells compared to the cell isolations. Again, so nuclei versus cell yields are going to be different when you're just trying to lyse and kind of pull out tissues, pull out nuclei from tissues. You're going to get more of them in a quicker amount of time. So this is kind of just a comparison of our data for liver, lung, kidney, and spleen for nuclei versus cell data. Not too dramatic, usually about a 50% increase. In the case of kidney, we are getting a lot more nuclei compared to cells. So, if yield is an issue especially for small sample sizes, plenty of, again, mentioning earlier our Nick Plus cartridge, plenty of our customers are doing samples of biopsies, and those are just tissue types that you would not really be able to easily and robustly isolate, enough cells for downstream applications from. But nuclei, you can get plenty of nuclei for downstream testing and sequencing runs, even from 1 to up to 5, those milligram small biopsies. So again, talking about this cell type representation, this comes from the same group. This data comes from us, but the notion of cell isolation by enzymatic dissociation, especially in brain, causes damage to neurons. Neurons are extremely difficult to isolate in cell isolations, especially in a quick manner, even for manual methods. So this is data coming from mouse brain cell isolation from the Singulator. Again, you can see a variety of different cell types that you would expect: microglia, oligodendrocytes, astrocytes, a variety of immune cells as well. And then there is a neuron population, a small neuron population, kind of right there in the middle. And comparing that to nuclei, again, what we see is equal or pretty much the same amount of feature RNA comparatively between the cells, and even lower, obviously, mitochondrial contamination, as we're not stressing out the cells as we're isolating them. But you can see, now we have this vast isolation population of neurons that we just, can't really isolate very easily from a quick and easy manual method. And kind of here's that full comparison, mouse brain cell type, mouse brain nuclei cell type. Again, you can see just the huge increase of neuron cell types being captured in nuclei versus cells. And this is also a benefit in the sense of, this nuclei isolation protocol especially for brain, is that 6-minute protocol. And on the Singulator and most manual methods for brain, it's about 45 minutes to an hour before any sort of downstream cleanup. So all of these samples also went through a debris removal protocol, which I'll go over a little bit as well in a bit. But a full-on 30-minute protocol for nuclei from start to pretty much start to finish to loading, and about an hour and a half to 2 hours for a cell type isolation. So here's a little bit of data as well from the University of Tokyo. They use our Singulator to do isolations of nuclei from heart tissues. And one of the issues they were having, again, with their manual method, was kind of high contamination of mitochondrial genes. They were doing isolation of nuclei by doing a small enzymatic digestion beforehand to loosen up the tissue, and then isolate by hand by, I believe, dowsing, incubating, and then usually doing a density gradient centrifugation. And with the Singulator, they were able to see what they had is they had to increase their threshold by a lot, and kind of take out a lot of those samples in their sequencing data. You can see the mitochondrial contamination was, greatly improved and reduced in the Singulator samples. They ended up being able to set the threshold, I believe, for our samples to about 2 to 3%, where in their old one they were setting it to 15% and losing a bunch of their sample. You can also see in the feature RNA that they were getting many more genes per nuclei in their data set than they were beforehand. So clearly a stressful method of isolating nuclei before using the Singulator. So again, some of the benefits of the Singulator for cells or nuclei are going to be high viability and reduced failures. Everything's going to be behaving the same time, no training or less training. Plenty of ease of optimization. Usually, again, as I mentioned, a couple runs of your type of tissue, and optimizing can get you a pretty good cell or nuclei suspension. It standardizes your upfront process that will help you reduce failures. Again, if you have precious samples and you're trying to figure out a way to isolate cells or nuclei from a small biobank of samples, you don't have to waste a bunch of them building up protocols. It enables again, flash frozen sample isolation, small sample isolation down to 1 milligram, and it's temperature controlled for both cells and nuclei. And it's open reagent platform. So if you have reagents that work well for your types of tissues, those are able to be run on the Singulator, reducing costs, but also allowing for less optimization. And then it's fast. So for nuclei on the S200, you can do 2 samples at a time. You can do your 8 samples for a sequencing run in about 25 minutes. Yeah, so the benefits, of automation, make everything, faster and more easily optimizable and more reproducible. So some other things I'd like to talk about are some upcoming things that we're excited about. So we're going to be releasing a nuclei debris removal solution. So one of the problems we hear from customers is obviously cleanliness of their solutions or of their suspensions for loading onto these single nuclei processing platforms. So we're excited to release a debris removal reagent, which I'll kind of go over in a minute. And then we're releasing a couple more protocols and reagents for different types of tissues, and that'll be skin, a tumor, and a heart isolation protocol. And then we're also releasing a large cell isolation cartridge for mostly for the isolation of heart cardiomyocytes, which can be up to 150 microns across. So kind of going over the debris removal reagent. Here's a little bit of fact sorting data of brain nuclei. So on the left, we have the brain nuclei before cleanup. So you can see a fairly, as expected, dirty sample, lots of small debris. When you look at the side scatter versus forward scatter, lots of small debris. That is indication of usually a lot of myelin, as well as plenty of large debris that's not stained with DAPI. Again, usually an indication of maybe a little bit of undigested tissue or plenty of myelin debris as well. So you can see about 39.6% of the total events before debris removal were nuclei, DAPI stained nuclei. And then after a quick, this procedure is about 15 minutes, a 15-minute density gradient centrifugation protocol. After this, about 90.3% of the events were DAPI stained nuclei. So a huge improvement in the cleanliness and of the prep. So as I mentioned, we're releasing a heart protocol. So one of the benefits, again, and that we want to be able to tell everyone is our platform works with a wide variety of downstream sequencing applications. So we've partnered and done some work with Parse Biosciences to make sure that our cells and nuclei work with their fixation protocols and with their sequencing methods. We've done work with Fluent Bios as well to make sure and to see that our cells and nuclei are compatible with their platforms. So working with a bunch of different applications and platforms is important to us. So here's a little bit of data isolating heart cells, and this is important for us and kind of necessary for us to do with Parse, as well as these cardiomyocytes can be very large and tend to not work with microfluidic platforms. So some of these cardiomyocytes can be up to 150, 200 microns in size and just not compatible with the microfluidic platform. So as I'm sure a lot of you guys know how Parse works, but it's split code combinatorial barcoding. So these cells or nuclei kind of turn into a vessel for adding the different barcodes. So the samples are loaded into plates, barcoded, taken up again, mixed up, put into a new well, added more barcodes, and then eventually each cell kind of has a cell-specific barcode. So it doesn't need to go through any sort of microfluidic platform or any sort of machine. It's all able to be done off of an automated machine. And what we saw was great data in terms of cardiomyocytes. Again, we were able to isolate a bunch of them, which we weren't able to do in microfluidic platforms. And again, a variety of different cell types, whether it be monocytes, cardiomyocytes, immune cells. And yeah, so the instrument was, we were able to prove the isolation of these difficult cell types thanks to parse. And one of the other benefits that we were able to do using parse is with the fixation of the cells, we were able to isolate from a variety of different tissues and kind of pool and do everything together rather than trying to slam everything through in one day. So that's another benefit. And that's pretty much all I have for you guys. Before I open it up to questions, I did want to mention that we are in the process of kind of getting a demo system out to you guys to get tested. And we're more than happy, obviously to include anyone that's willing and wants to try it out. If it's not a standard tissue type, or species type, like I mentioned, we're more than happy to meet and kind of make recommendations and kind of go through a pre-optimization. So it's, pretty easy once it gets there. Again, like I mentioned, things can be pretty easily ported to the Singulator.
Awesome. Well, thanks for a great presentation. Yes, if anybody has any questions, please put them in the chat, or raise your hand and we can unmute you. But I guess a good question. Thanks for mentioning the demo system. That's such an awesome idea, and I love seeing kind of demos come through. So everyone likes to see the data with their own hands and how it works with their cells. But I guess kind of paired with that, is there a rough kind of cost per isolation, like a rough number? I know it's going to change depending upon which specific protocol you're doing, but is there like a rough ballpark number we could have in our heads just for estimating costs?
Yeah, so on the I believe Hana, you could correct me if I'm wrong, but on the cell side, it's about $30 an isolation, $35. And then on the nuclei side, it's similar. The NIC Plus cartridges are more expensive. They've for those smaller sample sizes, I believe that's about $60 to $70 a sample as well.
Yeah, it's $70 for the premium NIC pluses, but for the standard nuclei and for cell, as Nate mentioned, it is $30 per cartridge, the single-use cartridge, and reagent could be anywhere between $5 to about $10 or so dollars. Of course, for cell reagent, you can use your own, as Nate discussed earlier.
Cool, thank you. Question coming in from an attendee. For single nuclei isolation, is it better to do it from fresh or frozen tissue?
Good question. I would say it kind of is tissue dependent. Usually frozen is better because it's easier to get frozen tissue than fresh tissue, and you can kind of have more of it available and do more runs. You'll also get higher yields from frozen tissues just because freezing, fractures the cells and makes it even easier for the nuclei to be isolated. That being said, some tissue types, really the only one that comes to my mind is like liver. Frozen liver nuclei can be more fragile than fresh liver nuclei. So you might see an increase in kind of leaky or ambient RNA in a frozen liver isolation. So that's kind of an example of one of the things that we do to optimize. So as I mentioned during the nuclei isolation protocol optimizations, is we have different degrees of intensity, and liver, frozen liver is one of the ones that we recommend doing at the bottom, the least intense isolation protocol.
Any other questions from the audience? No? Pat, any questions from you?
No, Alex, you kind of got at the cost question. That's always, the big question for the customers.
Yeah. We do have another question. It says, are the tissue dissociation protocols for human and mouse the same?
So yeah, all of the ones that we offer reagents for, so kidney, spleen, lung, heart now, skin now. I'm sure I'm forgetting a couple, but all of them are, we do the same protocol for mouse as we do for human. So those tissues are minced and incubated basically for the same amount of time and go through the same disruption profile. So I will have some customers who will take the chance to kind of optimize a little bit and shorten the protocols if they like, and try a different disruption. But out of the box, all of the mouse protocols work for their kind of matching tissue type in human. Do you have a protocol for pancreatic islets? We don't have a protocol, so I have worked with one customer who's tried to do this before. I think we don't have a protocol right now, and that's kind of one of the reasons why we developed that large cell isolation cartridge. What we were finding was the final filtration, just to kind of go over it, the final filtration sizes of the nuclei cartridges are 40 microns, and the final filtration size is 70 in cells. So that's why another reason why we developed the larger cell isolation cartridge was specifically to try and do work on pancreatic islets, because they're usually bigger than the 70 microns. But we don't have a protocol right now. For brain tissue, does it also require demyelinization process, or it just works fine? I think for the myelin removal, it does kind of depend on, from what I understand, your downstream platform. So and kind of how bad it is, I guess. So for nuclei isolation, I've seen and worked with some people who don't do any demyelinization process, and they kind of accept that risk of clogging if they're doing a microfluidic platform, and because they don't want to stress out the nuclei through another process. For brain cells, I do think it's necessary, mostly in my kind of opinion, just because the yield of cells is lower than nuclei, and there's just usually a much higher proportion of myelin to cells. But again, that kind of does have to do with the downstream application. So any sort of microfluidic platform, you're obviously going to be prone to clogging. If you're doing a platform like sparse or some sort of split seq platform, you're not at the worry of clogging. Why do you lose immune cells in nuclei isolation? It's not that we lose immune cells in nuclei isolation, as much as, or what we hear from customers and what we see in the literature is that it's more that it's easier to isolate immune cells in a cell isolation protocol. So lots of the other epithelial and endothelial cells are more embedded in the ECM, so they're just a little bit more difficult to isolate, and are more prone to dying through the isolation. So that's kind of why we see, and why most people see, a different population of cells when they do nuclei versus cells.
The next question looks like it's more for Pat. It says, if installed, would this then become an additional service option, and a lab could then provide frozen brain samples for something like single nuclei RNA-seq?
So, yeah, if installed, yes, it could definitely become another service option. Yeah. I don't, I guess the question is, is this something that we have sufficient interest in from the faculty to bring it on?
Yeah, that's another great application for the demo, is to be able to drum up interest and get everyone, in our hands, showing that that things are hopefully as turnkey as possible with a lot of it.
Exactly.
So I guess that's the next steps, is getting that demo set up.
Be to help.
Perfect.
Yeah, often that's the best way for people to get a feel for a new piece of equipment. Is if we can get several people to bring different sample types and try it out, then we can, in our own hands, and also the faculty who might be using it can see, if it's going to be useful to them.
Yeah, it's always great for us, getting the machine in, that, different sample types are run through it. People can take a look at the cells, take a look at the nuclei, decide, some people will even do sequencing runs, at the end of a demo or during the process of the demo, kind of get that confirmation that they're looking for.
Cool. And any other questions from the audience? Well, okay, so I guess we'll say thank you very much for a great presentation. We'll definitely be in touch about coordinating a demo so that we can try this out and see how it works on our ends. But otherwise, thank you for a great presentation, and have a great day.
Thank you.
Thank you so much.
Thank you.
Bye.